now in the fourth talk in this week, I am going to talk about cancer genomics. And there we will see some of the example where we have solved some cancer problems using what you were trained until now. And this, some of them will be connect you to the previous lectures. So whenever I go somewhere, people ask what is the best way to avoid cancer? And then I'll say, prevent it actually. So prevent the habit that put us on the risk. So don't use tobacco in any form. So don't say that tobacco thoda sa pan mein le liya, this is fine, and tobacco thoda sa cigarette mein le liya, that is fine. So avoid and let others also promote that they are not smoking and or using any form of tobacco. A lot like in Haryana, a lot of people still use it very extensively. When I went to Haryana, I was shocked that people use it extensively in hookahs and all. Not to use hookah also, and protect your skin from the sun. Do not overexpose yourself. Eat healthy diet. Manage healthy weight. Be physically active, and being physically active will keep you prevent a lot of other diseases. Practice safer sex, and avoid any risky behavior concerning sex, and get vaccine or immunized for viral vaccines, virals that cause cancer. And so the current coronavirus vaccine, which BioNTech has developed, actually these were for this purpose only. So they modified it very quickly and came up with a new vaccine. So if you have the vaccine, probably that will help in other viruses as well. So and obviously one has to know their family history and get a regular screening if you have thinking that you have a certain family history or certain problems, especially in women when they have problem issue with their especially for breast cancer. They see any bulges in the breast, so they should meet doctor. And early detection is also good, prevention is also good, so that one is much more safe. And then I'm not talking about the focus area, but then one of the focus area of our research is cancer genomics. And then what happens that mutations are triggering the tumor. So normal growth hormone will have, or suppressor gene will have no mutation, and everything will be fine. The cells will be growing very nicely. Normal cell cycle will be going normally in a controlled way. And when we have a mutation in tumor suppressor gene, leads into defective non-functional protein or poorly functioning protein, then cell cycle will not be under control, and we'll start getting a kind of problem which normally will not see otherwise in normal cells. So and that will lead into the cancer. So sorry, I'm going back. Okay, so then we already know that the human chromosome has 34 GB in size, and mutation on these chromosomes are leading into a major cancer. And so in idea of genomics will help, and using genomics will definitely help both in coding and non-coding region. And what people normally do in this area is to use multi-omics approaches, and we have seen it in past, but then I will explain you again that you need to have a knowledge of omics data, basic biology, clinical data, and pull it out using bioinformatics to a lot of information and fine-tune the data that will be used for personalized personalized healthcare, disease predisposition, understanding of genetic and molecular basis of human diseases. And so some of the omics, we have already discussed, but always better to give an example. The genomics, most of the people understand that understanding of entire gene and the regulatory element is genomics. Transcriptomics is or RNA-seq is the express genes or in a condition X. Not all genes are expressed, but a fraction of genes, and those in a specific condition express genes is called transcriptome. and a study of it is called transcriptomics. And then we have exomics or exome-seq, which is genomics only, but only coding region you are covered. And so if you have a study where you have less money, you can cover only coding region and try to see what you are getting. Proteomics is a study of proteins of living system, not DNA, RNA, but protein. So you protein metabolomics is a study of metabolomics. So proteogenomics is the combination of all. And proteogenomics will solve a lot of problems in cancer genomics. So this is getting more and more used to. But then you have to use interactomics, interaction between nucleotide, protein, and metabolomics, and then connectivomics and pharmacogenomics, phenomics, physioomics, exposomics, environmental role of environment overall, and bibliomics. So a lot of literature has to be combined with your data. Extensively, especially in cancer, you need to know each mutation, role of each mutation is important. So there, if you have tab bibliography with your results itself, it is much more easy to study than tapping afterwards. So how this is started? This starts with the whole genome sequencing getting cheaper with time. Earlier, when the human genome came, the human genome costed billion of dollars, and then next generation sequencing come up, and then today you can sequence at your in Bangalore, say 70,000 rupees. You get your genome sequencing done, so less than $100, and even cheaper if you negotiate it. I'm telling you retail price. And what I have seen is the $560 minimum we have sequenced a genome. So in for the research purpose, and then both cost of sequencing is cheaper and cost per megabase pair of raw DNA sequencing is cheaper and cheaper. And this is surpassing Moore's laws, which says that with time, technology grow not so rapidly that it is not going in a linear order. So in the bottom, I'm showing you the mobile phone data for 3G, 4G, and high performance computing and all, and they are following a linear trend from 1995 to 2020, not a zigzag what a human genome is seen. So earlier people had the human genome, people were thinking what to study carefully so that we can cover in that budget, because budget was very high, and this is very useful. So people sequence human genome. Today you can sequence anything you like because it's so cheap. But then question is why you want to sequence, and the question you have to ask other side of the question. That time what you want to sequence to sequence. Here you can sequence almost everything, but then why you want to sequence, and that is the cost. But then data volume is also doubling because of this cost. Data is also doubling in every 6-7 months, and this is a story of initially 6-7 months was picked up in 2015, 16, and 20. By it's every 4-5 years, 1 month shorter. So if there was 6-7 months, probably now data is doubling at 6 month basis, and data is as big, and there are different kind of data: genomic data, insect data, earth biome data, data from plants, 10K plants efforts, 5,000 insects. So not just human you can study anything on the planet, and then a lot of human and cancer data is already available, so that if you do not sequence anything, you can still, if you know computing, you can actually use that data for next 25 years and happily do your research and maybe training, whatever you want to do, because there is a massive amount of data. Big initiatives of the governments are getting, like India is also now in the list, which I have not listed. I wish should that India is also sequencing 10,000 human samples, and so that this data is one of the biggest data for the comparable, much higher than YouTube and Twitter, and close to what is data acquisition terms, slightly less than astronomy, but data storage, which is required, is 2 to 40 per EP per year, exabase pair. And this data is heterologous, requires a lot of CPU. CPU uses data can be as small as 10 MB and can be as massive as 10 TB in size, and you want to require a good amount of data. You need to manage it properly. You need to you have a lot of opportunities within the data. So because data to knowledge transformation, as I told you, so there is a massive use of these data. And as you see that not even YouTube, which we are using frequently, is capable of producing and storing that much data. So in the advent, people started doing the cancer genomics, and a lot of people have written review. Another recent review by Peter Campbell and Jyoti had just arrived, and then they are saying genome sequencing to journey through the cancer, where they discuss, and probably you are aware also, but I can explain you. The cancer can be of two types: somatic and germline. Somatic, which is mutationally acquired, and then by doing genome sequencing, one can find driver mutations, one can find chromosomal aberration, and then one can also report mutational processes, DNA repair, ultraviolet aging, with what is it related. Another type of cancer aspect like tumor heterogeneity, telomere length, and if one is also covering germline, also tumor length can be detected, family risk, drug metabolism, and several other things can be covered. And we have extensively focused on the familial cancer risk and cancer in family. So we'll show you good examples later on. And this can be then used in cancer management, and also understanding how cancer is, how clonal expansion is happening in clinical and diagnostic and classification of cancer, outcome, therapy, resubmission, advanced relapse, all can be understood with help of using genome sequencing. And therefore there are massive role in the cancer, and there are several roles who have been studied. And so when I'm saying genome sequencing, I'm actually meaning sequencing or resequencing of human genome. Because a reference genome, one healthy individual genome is already known in databases, so that's called reference genome. So that guy is there on the left corner, and then you have a population have different traits on the population. So you can compare, sequence again and compare with the reference, that is population genomics. When we compare the cancer patient with the cancer samples, different cancer, I have it idea is that in different colors. The cancer samples with cancer genomes, sequence the cancer genome with and compare with the reference, then it is called cancer genomics, and so on. It can be applied to any disease, like say cardiovascular genomics is the study of cardiovascular samples with respect to a reference sequence. And so this is what is sequencing. And then when you are sequencing in a layman perspective, what is the meaning? So between me and anybody in the world, if we look different, there is the one amino acid or one nucleotide or 5 nucleotide differences are enough. So if, for example, I have taken AAC in the reference genome at the positions in population, I see that many of the population has this reference, but then one of the individual has AA is changed into CC. And these differences can tell several traits of individuals plus several traits of population. Say population of India is different from population of China, or population of Bihar is different from population of Madhya Pradesh, or so on. And so these data are placed in the 1000 genome and Genomad. And one of the Indian study which I remember is that had the origin of Indians have been covered by CCMB in this aspect. Then the cancer samples, same mutations can be checked for different cancer, and these are reported in the in the Cosmic Bioportal, C-Bioportal, and Icgc. So we will be seeing these mutations, and we'll be also seeing the some of the examples of the databases later on. And but before that we go, so basically we will cover some portion of theory and then some our own examples. So what happens when tumors are sequenced, you will get results for different tumors. You can get by genotypic information, like which amino acid, which nucleotide is changing into which nucleotide in which sample. And also you will get regulatory genomics with non-coding area and missense mutations, and pull this missense mutation onto the structure, and you see the number of the protein structures getting higher and higher. So one can pull it out on the structure and see a structural implication of a protein mutation in the cancer. So, and this will help in the realization of personalized medicine, and can have implication of using a role of genetic variation into drug response, drug efficacy, toxicity, and so on. And that can be studied from the non-risk with risky carriers and all in the general population. So one of the example here I'm taking of multi-omics personalized. So in a population, you have different cancer and normal tissues. From a person, you'll take it, and then you'll do a whole exome sequencing, RNA-seq, mass spectroscopy, and predict immunogenicity. And then you combine different experiment, and you will know what is the region for which mutation is the region. Once you know the mutation, what is the if these are kinases, there are kinase inhibitors which are involved, and you can give, if the mutation is in kinase, you can give a kinase inhibitor to treat the cancer. So, and these are as well set of kinases, known kinases with inhibitors. Right side is given. And if you want to draw this figure yourself, you can use Yasara, PyMol, and Rasmol for structural modeling tools. Then you can draw such similar figures. And that's what I have named it here. But most important was one of the study of personalized medicine, where the breast cancer samples are done, and samples were first sequenced by NGS. Then the computational analysis identified into three groups actually. One is a HARP mutation of V77 and T798, and PI3CA V1047, and ER mutations, three different gene mutations. And then you look for preclinical drug risk screening. And what you see here is that they will have a different drugs one can give. So they come to one has the HERP monotherapy, one simple drug is given, another one you have both a drug is given for anti-hormone therapy and PIK3 inhibitor. A combination of drug is given, and second sample again if can be given a drug of HERP inhibitor and PIK3 inhibitor, and then third one is the again anti-hormone therapy and HERP inhibitor is given. So basically 3-4 combinatorial drug is available for group of people you want to treat, and then you can decide which group and which person. If a person has this HERP mutation, will go for HERP therapy. Person has PIK3 mutation, will go for any of the treatment, and then person with the fourth group, ER mutation and HERP mutation, will go for the fourth therapy. So basically you get different therapy based on the mutation which is existing in the patient. So given that, we have also developed the pipeline for such analysis, and shortly I'm giving you a hint that you combine proteomics, genomics, and RNA-seq. So you can do independently and then combine the data, and then this can be a block: genomics, transcriptomics, mass spectroscopy. Merge the data set into databases and use the cancer data sets and pull it down to this this sequencing, pull it down and make sense of the data. So this I think this will come in 2021. There is a delay in the review to be revision, and then it will come out on the public. So in this, what happened that in a proteogenomics experiment, you see that DNA variants are so high, RNA variants are so high, somatic variants are so much, and proteomics variants are so much, and only few are some more here. And sorry, what you see, and then splice junction variants are again studied, and proteomics based junction are here. So when you look carefully, you see this these mutations are predominantly white regions are where they're not matching. So this, suppose this 3 experiment in GATA3 is matching, but the one of this has a gap, so this is not covered, and so on. When you look very carefully, actually one can map down several of these using using the using the proteogenomic method. So basically, you will see the gaps, but when you combine with proteomic experiment, one can fill those gaps, and that's when the proteogenomics will is required, because single omics will start failing at one point during a complex scenario like cancer. Once you have the RNA-seq proteogenomic data and global proteomic data, you want to identify which network they are in, and these are the papers which are different ways of doing, and then finally plot your driver mutation to the pathway, and then study the cancer and finally make a hypothesis in solving cancer or resolving against any issue. So these are different papers which combine pathway to driver mutations or so on. And then what we want to do today, and what we normally do, is that we take out the data from cancer genomics, and then and population genomics. So GenomeAD is a population genomics data set. And then you have TCGA data set, which is available at the different data set. We'll go to them and we'll play with this data set, but these are the major data set which is available. Icgc is International Cancer Genome Consortium, is from different cancer which are prevalent in different part of the world. The countries are spending their own money and they are pulling the data together. And one of these data is actually coming out of India, is the oral cancer. India is spending money on in this consortium, and so that these data provide you with large genomic samples, mutations, and other data, and that will help in resolving cancer in a long way, not only for the country specific, but also once the data is fully available, anybody can play and make a hypothesis and find the results. So what we have done so far, or I'm going to present, is that we have studied in the families. So what we have done, that we have identified, we have taken high risk cancer families, and that done sequencing of more than 53 cancer families and 257 genomes we have taken. And then what we have done is that we have taken these families, the colorectal cancer, cervical cancer, Hodgkin's lymphoma, multiple myeloma, pancreatic cancer, PTC, thyroid cancer, actually, a small intestinal carcinoid, and so on. NF stands for familial, because we're coming with the family history. So if in the family history, this is how this family pedigree will look like. Male is a square, and woman is a circle. And then you need one who have the same color. They having same type of cancer. In this case, Hodgkin's lymphoma is red color, another color leukemia is orange, and so on. And then three families I'm showing you, and this all starts with the genetics. The genetics play a role in human diseases, and one of these on using this hypothesis that there was a Time magazine covering a kid. Do you want to know my future? So parents want to know their kids' future for sure. And this genetic counseling can use if they have a knowledge of these mutations are existing in this cancer, that cancer. If the kid has, they can go for it. So in this regard, we have developed a pipeline. Initial pipeline was only for exome sequencing and uses missense variant and variant 3' UTR and 5' UTR, and then was exomic. But then the second version of the pipeline uses 12 deleterious variant and 5 intolerance score, and then uses a 5' UTR, 5' UTR plus all other non-coding segment. And at times, this was also published in 2018. At times, even you use both the methods, but you cannot resolve the problem finally. So you will do a pathway stratification in this regard, and that will become available available to that will become available to us in the pathway, that which gene is responsible in the pathway, which we've identified, which mutation of that gene is important in a pathway, and can we say something about the gene that we have identified. So what we do in this process, we sequence the genomes of the we sequence the genome of the case and control from the family, and then we call the variant. And from after variant calling, we collect the variant and annotate it. In some cases, or in most of the families, we take the such calling application, and we call the SNVs using GSNVs from GATC, and from GCNV we use CNV annotation of genome structural annotation using VNC annotation tool. And then what we do is that we call the variant parallelly and filter the variant for the quality. You don't want a bad quality variant to be called. Coverage has to 5x, means and minimum 5 reads should cover on the analysis. Quality score has to be 20, and minor allele frequency MAF, because we are studying in family, so the variant will be rare, because it cannot happen that this is causing cancer to population, because cancer is limited to the family. So once we do that, we can take those samples to the pedigree, and in the pedigree that okay, these cases will be allowed to have variant, these cases is not allowed to have variant. Obviously cases will mostly allowed to have variant, but then there are cases where we will not allow, and depending on pedigree information. And then controls should be allowed, controls should not be allowed. And then we do first round of filtering using CAD score. CAD score 10 means top 10% of human mutation, top 20% of human mutation, top 30% of human mutation, and so on. And these are scores for mapping. A score for mapping most critical mutations in entire genome, so that will make the number of hits lower. Then you will only work with the good hits afterwards, and then you classify this into coding region, 3' region, 5' region, and non-coding genome, and use a set of tools for that. But then in genomic region, the most of the tools will fail in non-coding genomics. So you have to employ certain other criteria, which will explain you next slide. And once we have that, we will place the variant for visualization and look for potential potentially good variants whether they are found in CleanVar, whether they are found in human protein atlas, whether they are found in BioGPS or they are found in DG's genomic databases, OMIM, and also if the mutation is in a germline variant, mutation libraries like Canva, CBioportal, Cosmic, Icgc, endogenous for somatic mutation, and we'll manually also check the mutation for confirmation that whatever we have identified is should not be in a problematic region where by which we cannot give a control. We people will start doubting our result actually. So it should not be enough problematic because ultimately you are solving you are helping to solve a disease. So we can't say that theoretically it is possible, but there is a genome sequencing error. We have to be very firm what you are saying, especially in the cancer genomics. In basic biology, we can even towards end we can say if but here there's no if but because either you will save a person or person will die. So in the non-coding region, we can use super enhancer and promoter regions, and then BET tools intersect function will be used for finding the critical mutation, which is where our mutation is in the super enhancer promoter or or enhancer promoter region from phantom 5 or not. And if so, if they are found, they can be considered critical, and then they are placed on the transcription factor binding site, or whether they are not there or not. Then we'll use a CAD system for seeing whether they are on the uh, chromatin changes or not, segmentation is going on or not, and whether they are in ultra conserved region or ultra sensitive region of human genome. And if they are passing one of these four criteria, we'll put them into top ranking, check for the expression, RNA-seq, motif change, and other regulatory element. It will come back to the similar pipeline like what I have shown previously. We'll see some examples of our work. In one of the thyroid cancer families, we have applied this, and in thyroid cancer family, these two patients should have the mutation because this is common cancer, PTC, and the third person has another phenotype called micro PTC, and then micro PTC is considered another disease. So we taken this out. Only these two cases should have mutation and allowed, and then we did the whole genome sequencing, and we identified initially by variant calling annotation and minor allele frequency. We identified this much mutation, and then we had a pedigree segregation in this. We have a pedigree segregation. After pedigree segregation, we identified 1970 mutations, and there's 97 70 mutation. 95% are in coding region. You see here more than 95. But these are next step are not able to predict any promoter or enhancer in their area. So we only look for those less than 2%, which is 28 out of when we applied CAD 10, we are left with only seven mutations, and out of 7, only 5 are useful. And now because these 2 are in UTR region, so the missense will be considered. This 5 will consider further. 4 missense and 1 non-frame shift. Non-frame shift is also out, and now the missense variants are taken into the consideration, and we chopped 3 of them out, and only 1 was left, which is found in CPXM1. And when you're saying CPXM1, that means something. Why this one? Because these are the green ones which are favoring that mutation will stick to the mutation. So CPXM1 is one which is left out at the end, and we saw that this many are left out in the G573R is left in the or with this mutation is found in the peptidase 14 M14 mutation. So the step by step we narrow down and we end up with a one mutation in the family, which is critical. So when we are saying the critical family has a critical mutation, and then we have to look into the further look into the protein alignment, multi-species alignment we have made, and then we realize that this is a very conserved region where the mutation is mapped, and this is a critical mutation because G is very predominantly found in different species, and R it is changing into R. Though this gene was not previously known in thyroid cancer, we identified first time a mutation in this gene, but this gene is actually tumor suppressor gene in breast cancer and some other cancer. So therefore, this will have a role. Potentially, yes, indeed. And then but this is not very strong cancer gene. So when you are going to publish it, your study will be the first one. So that there are many people who will doubt your study. If you are the mutation is in the very famous gene, people will not doubt much. But if it is in a not in a very famous gene, people will doubt. So 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 the then we need to go but in life in actuality it can be anywhere, yeah, right? So we don't we're not biased, but we'll show you some good examples also in here, which is one of the mutations we identified in Hodgkin's lymphoma in dicer1, and then second one where we pull everything in a pathway context. And as you see that Hodgkin's lymphoma is a cancer of lymph node, and the pipeline and all I have already explained to you. But I wanted to focus as is a cancer of lymph nodes, and we have three families where the cancer is found, and the red ones are the cases, and then these are these are stars are those which are providing us which we have done whole genome sequencing. So when we combine this data into again into the pipeline, we get some numbers. We did the segregation. We again get few numbers, fewer numbers. At the end, we've identified 5 genes which are critical in the first family. Dicer1 is one of them, 3 in this family, and 5 in this family. And then what we did, pull this into the pathway context, and we see that hematological diseases, immunological diseases, developmental diseases, endocrine systems, and RNA post RNA modifications are critical, which are indeed true for this kind of cancer. And these are the genes which we have mapped. mapped, and then which are in peach in color. And then from one mutation, we using one mutation strategy, dicer one was deduced, and this was found in the cancer versus cancer versus non-cancer. So so uh, so this takes us to come to dicer, and dicer, this mutation was in this side. And if this gene is indeed responsible, this gene is involved in microRNA regulation. So there has to be role of microRNA associated with it. So what we compared with it, that the wild type gene versus mutant gene will has a change of expression pattern. So these are the different expression pattern compared to these ones. And we could see that there is a change of expression pattern in the samples across. So these are the two samples are upregulated, slightly up and down, slightly down, but the other two samples are always down regulated and heavily down regulated in last micro with respect to last microRNA. So that shows that significantly down regulated in the families, the members, those who carry mutation. And then this is not happening in a way random fluke. We just tested this with these two members, and we've identified this is indeed have a reduction in function. So that was applied into non-Hodgkin's lymphoma. Then recently we have published a mismatch repair gene mutation with large duplication. So so what happens is that what happens is that they this study will provide a new mutation in MLH1 gene and some other gene involved in Lynch syndrome. So what we identified that 3 families, we sequenced 19 such families in our pipeline, and we identified 3 families are carrying a mutation in different mutation. One is the large duplication of the family 1, where 2 cases and 1 control has a very large mutation for before exon 4 to end of exon 14 is duplicated, and it is forming a duplication is forming a disruption of gene, and this disruption is causing frame shift in the protein region here. But at the same time, this destroys the gene machinery, so gene will not be functionally active. And then second example, the second example has the MLH1 frame shift mutation in the two of the Lynch syndrome, and one is the endometrial cancer plus colorectal cancer. And so these 3 were sequenced, and we identified mutation in these 3. And third one is identified into 3 samples as well. And then when I said we are looking into genomic sequencing, I'm showing you that these 3 samples have the mutation. I'm confirming at the end. So the 2 males and 1 female. So these are the 2, 1, and plus 1. So each time we are looking into it, and this is the largest. This first one is the largest mutation that is known so far about these genes, and these all are new mutations actually. So we tested this with the immunohistochemistry and microsatellite instability test, and we could identify such large mutations are actually indeed true, and these are caused by these are novel mutations. So these are direct candidate for uh, directly pushed into inside database and directly use in genetic counseling, because the genes are already in genetic counseling, and novel mutations will go into genetic counseling. So with this approves what we are doing. If we hit to a top gene, which directly has implication in human diseases. Plus, the new ones will require much more scrutiny and much more carefulness towards it. And then we come to the training ourselves. So we have a pipeline where we have used similar strategies, and what I'm going to pipeline is available in immunobiology if you want to read. But what we are going to do is that we have in this paper we have identified several mutations from several databases in a single gene, and then these mutations are plotted onto top mutations within a gene, within one gene in a cap for cancer. And these are the mutations. So you already know how to plot sequence logo using muscle. And then we have plotted this on a structure using SRF PyMol. This you can do it, but then suppose you want to identify the mutation. So this one will be having practice in a genome ad. We'll give our type our gene and look for the mutations which we have recently published in today's spectacle. Okay, so Serpin H1 or HSP47 will write, and then these are the top mutations in this paper in Genomics which we have recently. So look where are these mutations? Can we identify these mutations in different cancer genomic databases and normal databases, and then how these are visualized in those databases. We have gathered it from here and analyzed it actually further, and we identified combinatorial 20 mutations. And can we also look into a genomics data set in the example database, so that you will have a feeling how the data look like, yeah. And then we can also do the same with the cosmic analysis, and if we can build some pathway for a pathway also. And then you want to reading material, you want to start reading about genomics, cancer genomics, you want to use biostar, biostar as a platform for discussion, bioclues for discussion, and sequencer for discussion. Plus you can use biolinux if you want to start linux using, and there are several books which are available for learning Python computing and a bit of bioinformatics. Yeah, again I thank you, and then we'll go and do the experiment. So I'll just go back to this page where we have listed a number of variants. Okay, so we are going to take this variance from this slide and going to start the analysis. Okay, so if you have a question, we can have a small break, and then we can start doing the experiments in some time. So if you have a question, we can have a shortly answer few of them. So okay, so I see that okay, so let me come to it. So we have done so many things in the course, so let me close this. Many of things which you, yeah, so let me leave this, and so that otherwise we have a lot of windows open, and lot of windows means confusion. Okay, so let be here. So you need 4 or 5 windows. Okay, I've taken 4 windows. You can also have 4 windows, and I'm giving the Icgc this mutation. so that he can transfer it to you and you can start looking. Okay, good. So let's start with Genome. Genome AD database. Okay, I'm taking it. So it will Genome database. Okay. Okay, so we have the list of the genes, so we can have a look. So I'm going into Genome. Just a minute. And I'm saying SERPIN. This is called SERPIN H1. Yeah. So this was published in our genomics paper, and now I have SERPIN H1. So what Genome is providing is that it is providing genomes information. How many synonymous mutation this has identified, how many missense mutation it has identified, and so on. So what we are going to do is that we are going to look into this mutation. So when you go here, this is showing the exons, introns, and showing the different mutations. So missense mutations will make sense. Loss of function mutation will make sense, but this non-synonymous mutation, synonymous mutation where there no amino acid change will not make sense. So I've just taken it out, and all other mutations also, like intronic ones, I'm taking it out. We are going to focus only of those two coding mutations. Okay, so you see that when you go here and you see you are going here, suppose you are here, you're here. So what this one is saying, the variant ID is the chromosome 11 position this. There is a deletion which is changing into T. TGC is deleting into T. That means T is available, GC is not available afterwards. So there is a way of writing it. Is if but coding one has only one last codon is changing because the C is changing into T at this position in chromosome 11. Okay, so what this is showing here, you can have a look, and if you move towards this, you can also let me have a. Oh, I need to zoom out. Okay. So if we are here in the, suppose we are here. So as we go here, this region means we are at somewhere here. This region means the table will go up and down. So basically it's mapping with respect to the table here. Okay. So as you see here, that viewing in the table here. So this table one is, so this much is visible. So when I go there, so you see the bar is moving here, which you are viewing on the table, whatever you are viewing on the table. So and these are transcript information, all information is given. Now what we want to see, that one of these mutations we have, which we have mapped initially from this only, can we identify this mutation in this? So first mutation is 76 and second is ARS to cis. So let's see. First mutation was 6, not it, P dot means protein mutation, and then the position is given. So 76 was the position. 76, yeah. Serine to leucine, yeah. 76. So let me see if this was the mutation we are looking at. Serine is there, but not to leucine, but tryptophan. Yeah, but this was there here, right? So you see. So what it says that this critical mutation we have identified, and this is found in 3 alleles out of so many alleles. So it's a rare mutation. And now we can click this mutation here and see the implication. So this is say that it's a very rare mutation found out of this many alleles, only 3 alleles, so frequency is very low, and so it's a rare mutation. And then this mutation is probably damaging and deleterious, and if I just look at into different ways, this is predominantly deleterious, and therefore we also have found it. And then here you can check if there is 3 alleles, which type of population has the allele. So European population has the allele total, and the males, they all were males. The 3 were males, and this is also providing an age. 2 of them are 40 to 45 age group, and 1 is 60 to. So population stratification and age stratification and gender stratification is also possible in the from the population data set. And now then we can see the mutation in the population data set. Okay, so the position was 621. So this mutation we're talking 621, not the other one. And then we seeing then the sample, many of them had G, but then many of it don't have. So C is the default one, and G is the variable. So you see that. So in the sequencing, when you're calling variant, so variant calling is done like this. That you see there's no variant here. So what is happening if A is present when wild type all have A, then no variant. This is not called variant. But C is changing into G, or G is changing into E, whatever. You have C is changing into G. So what happens that C is the default. Many of them will have C, but 40% or 30 to 40% have to be alternative positions. Then only it's called variant. Not like 1 out of 20. Then it is variant. It has to be substantial statistical support for other variant. Otherwise, it is considered error. So now this is inbuilt IGV. We use IGV locally also. You can download locally also and run. And this is inbuilt one. So basically, one can run it. And that's when you will find it, and that's one of the variant which we identified in one of our database, and we try to use the different data set. So this is one. Then we have used Cosmic. But you need to practice it, yeah. So just give a practice. So now I'm saying HSP47, or what Serpin H1 is called. Let's see if it is not getting. So it's getting Serpin H1. So I'm just taking it, or I can just take this one. So what do you see here in the? This will give you gene view, overview, external links, drug resistance, tissue description, genome browser, and genomic mutation distribution, variants, and references. So first one is a gene view. When you see the gene view, you will see these mutations substitutions are available at these positions. If there are other, so what is known about the gene, what is the different names, blah 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 blah, a lot of things. You can go and play. What is the drug resistance? Obviously, this is not a kinase, so drug resistance and drug is still not given against it, I guess. So therefore, it's not shown. And then tissue distribution, which cancer tissue has mutation. So you see there's different ones. So this gives an idea what type of cancer will have where this gene will play a role. The higher the mutation in the sample, the higher and the genomic location and all is given here. And we keep on going. So I'm just accepting. Okay, and then type of mutation distribution. So you see that missense variance is 45%. Synonymous is 29%. So synonymous will not make any difference. Missense will most effective in frame shift, insertion, deletion, and so. But then these two only are a big category here in coding. And what kind of substitution? Where A is changing into C, A is changing into G, A is C change. So the kind of pattern of mutation is also shown. And then these are top mutations. So when you are looking for one of this, so this only entry of 110 only. Now it's just a entry of 100, and of course you can export this information. Okay, so now 76 S2L is found here, but not S2, the one we have identified there. So this will be not so critical. We have seen there only also. And then we have in our paper, we have, and obviously we have top 20. So 103 R to 16. Can we find it? 103? No. It is not present. So in this database, we have to look for another one. So 116, and then 116. This is also not present. 159, 156. Serine to 159. Serine to phenyl. No, this is also not present. So one arginine 280, and let's see. Arginine 280C. Arginine. Yeah, so this is present here. So one of them identified. So with different databases we have. Yeah, so we can have a look here. And this one gives again the information about it and which cancer it is involved, what kind of papers it has, or if there is a paper on a study. Here author journal is not there, so it's not published in as a paper yet. Novel mutation. And then yeah, so pancreatic tissues, and so the from mutation we'll get a lot of information. And this is pathogenic, and we have shown pathogenic means critical. We have identified already from our comprehensive study. So and then which and then one can also identify from C bioportal, which is carrying all the largest data set from all other cancers. So let's see in C bioportal if TCGA data is also available, primarily available from here. C bioportal is the website of genes. So now we can take all, so you can say all pan cancer, 32 study, and then I want to look for genes. So SERPIN1. And this will take time because this is very large study. So it will keep flipping and finding. Okay, so what we see here is that this is giving a hint that this gene is involved in esophagus cancer with more than 5% mutation. So that's very significant. Ovarian cancer, and also more than 5 for myeloma. So this gene is definitely involved in different cancer, is heavily mutated, say at 4%, 5 different cancers. So this data set is giving you different plots, mutations, all different kind of data. And now I just wanted to see whether this is giving a critical mutation which we have identified. Okay, so this is giving lot of information. So 280C R into C. Okay, so this was required. This was found in head and neck carcinoma. In this sample, this is a sample ID. So we can go here and study what this sample is doing. What kind of other mutations this one has. And one can also look into this mutation. This is missense we have picked up already. Frequency is 44% in the sample. Sample 173 has 173 samples. For head and carcinoma has the mutation. So this is good mutation. So we can also look for one more one. So I'm just guessing one of this 339, 773, 777, 399. So these 373, 377. 373. Okay. Yeah, so R is changing into P. Let's see, R is changing into C. I'm checking out R is changing into C. 377 is not present. So 377 I'm looking. Okay, so this is 26 mutations only. Okay, let me show the all. Then I ask this question. Okay, it's 377P is there. 377. No. 339. Yeah, so 339 R is changing into H. Let me see if we are the same one. No. So R405, can we find 405? 405. So 4. Yeah, so this is giving some of them, and some of them are not there. So you combine. This also shows that the combinatorial effort has much more implication because you are getting critical one from different angles, and then some novel ones which are not until now reported. It might be available in Indian sample, which is not available here. So we saw that one can employ, and if you know your mutation, you can start doing it. And let me give you some more. Yeah, so then these are the mutation, then which pathway this one is involved. This is also giving an idea. And this one is also giving an idea of not this one, but I'm just interested in this one. Pathways. Okay, so this is the pathway they are saying. And then you have some mutational other plots down. You can download the data. So if you play with the data sets, you can have a look, and then we can also show you the ICGC maybe. ICGC. So ICGC data policy is different. We need to download it, create an account, and download it or use it. I think so. Yes, so this ICGC database is ICGC. Okay, so ICGC dataset is one need to create a login now because it's not freely available, and but this is available from C Bioportal. Uh, but for an individual to download, in one need to download it. So basically, you combine the databases and you can use. Let me see if something is lacking. We have not done. Okay, yes, pathway we want to do. So we can use the string pathway tool. String protein interaction is covering a string DB, and you can use your protein, maybe your protein of interest later on. But what we can do is that we can say HSP47 shock protein I'm giving, and it should work. So it's asking which species you are looking for. Okay, I just say human. So it's providing a graph with collagen. So all collagens are binding to this. That's true. But one can set it parameters that you are looking for full network, you are looking for confidence score, confidence or whatever. So I'm giving a confidence, high confidence 0.7, and I am saying that I'm interested in more 50 or more indicators rather than 10 or so. So default is still showing less. So now you see a lot of other genes involved are also shown. So this is the gene we're looking. A lot of collagens are binding. So you are getting a path protein interaction database. So what I can do that we can export this data. So what is this data is all about? So let me export it for 1 minute and I will show you why I'm exporting. So this gives an idea that some interaction is happening. And this is 114. Okay, so let's download protein sequence. There are protein mutation. I'm just downloading one file, so that will open here as a Excel file. So this is showing that this many genes are there. Okay, so I'm just taking the node, and I'm just taking this to. So there are a lot of genes, lot of. So I'm just removing duplicates, and removing duplicates. Okay, so there's no duplicates found. Okay, no problem. So there are 52 proteins are interacting with this because we are given 50. So I'm just taking it, and one you can do is that this network is one of your network, which you can obviously use export as a file, high resolution file, PNG, high resolution. Okay, and then what we can do is that we can go one more experiment, then we are finished. Enrich R. Then you will see a more path. So this is more pathway like enrichment. So we have seen that we have list of 52 genes from the category from there, and I'm just giving them into enrich R to see which pathway is actually involved in. So there's a lot of path. So I'm seeing in pathway. So what it is showing in the which in bioregulator of bone mineralization pathway, platelet pathway, and if I look at the cancer scene gate mediated signaling pathway, reactome is saying collagen biosynthesis and modification pathway. So this is good one because this is collagen forming And then when you look into collagen, reactome say clearly, reactome is saying what is the p-value. If you generate table, you will see what is the p-value and all. And then here you see that how many genes are involved in this collagen biosynthesis. They all are shown in this table. You can extract this table. Then you have collagen again, extracellular matrix. So you see that we can build a take from one tool when construct another set of pathways. So this is indeed true that HSP47 is involved in collagen biosynthesis, and the component binding here I have taken here, and it shows that more or less uh, true. And you can take more than one databases, say look into Panther, what is happening, something some integrin signaling pathway, something close to the function. You will get it from different pathway depending on what is the information pathway is gathering, pathway database is gathering. Yeah, so with this we end up, and hope you are learning. And it do not start I do not start building, and it was not never a one day work like this figure. Suppose it took me several days of strategifying, and then we decided, okay, we go for this figure, not one day job. Okay, so let us wind up for today, and if you have a question, please ask me. Okay, so Siddharth Singh has something to ask, and somebody has raised a question, so people can raise a question. I'm seeing I'm not able to see the question actually, so you can switch on your sound and ask your question. Because I'm seeing the one participant hiding or something is going on. So that's not a problem.
Sir, one question we have: how to extract miRNA from Icgc genome sequence?
Any EC genome sequence.
How to extract miRNA from Icgc genome sequence?
No, TCGS or Icgc genome sequencing. So these are you know the reference human genome, right? So they have microRNA. Suppose I have a microRNA here. I want to look at the microRNA rather than I said look into the cosmic, and instead of saying gene, I can look into microRNA. Let's say MIR 122, something like this. Suppose this is MIR. Then I will start getting. So you have to find, let me see, microRNA number. I normally don't remember the numbers actually. MicroRNA number. So this will give me a, suppose I have a MIR base, yeah. So I go to the MIR base. You can get a human HSA 121. This is number, yeah. Or so you can go and look individual also, because in a human already defined microRNA is there. So you can extract information as I do it for gene. Consider microRNA as a gene. You will be able to do it, and then if you are able to extract all the genes, then you can same way you can able to identify all the. See, there will be some trick, yeah. So 121 microRNA is same way extractable from any database. Maybe we just wanted to show you one. Extractable from any data source. Say, like I'm saying here, which microRNA we are on to say let 7. Yeah, let's see. Let 7. LET. Oh, it's yeah. Let 7. Okay.
एक बार हम शेयर कर देंगे 5 दिन की अटेंडेंस अब एक बार कंसोलिडेट करके
we'll start getting microRNAs also from the data set when we put exact detail of the, so maybe microRNA database. my RNA database, my RNA DB. Suppose we have a database, Mirbase, we are going to Mirbase. So you can browse or microRNAs from here. Okay, so these are names, right? So this same way, same thing can be extracted from different databases and combined here. Where are the location? What are the information available? So like for microRNA, but for microRNA, always remember microRNA themselves will not make sense. They have what they are regulating will be more important. So in the last lecture, as we did, we took the gene and then looked for the where the microRNA is binding. So that way we can combine with the cancer. And lot of and then also there is a somatic microRNA database. MicroRNA. There is a database called for somatic microRNAs. Uh, yeah, somatic mir database. From there you can take the information directly. So yeah, so that's you Google this word, you will get it, and then combine the data from the different databases. So there will be a definitely a way, a trick. I'm telling you that all the information is can be gathered. Gathering can be easy, gathering can be difficult. So now they have this search, they have download, they have all the options. Okay, you can download from here. So all the cancerous data set is provided here. And then they have done some experiment also. So now you are able to see, right? And yes, so that's when like no will be so straightforward. We always have to be always have to play with the certain tricks. And once it's straightforward, it becomes and then yeah. So suppose so then this experiment micro this is a different data set actually. So now they have provided already a data set is provided here, and again you can let me see. Yeah, so you're getting a lot of information from here itself, and yeah, so this is mature RNA. Maybe this one can. So it's a question whether the database will allow us to search in which way. Let me try this. Uh, uh, so okay, so there's different way, and this time is fine. Okay, so this is microRNA. We can now extract it. So let's see what you're getting. So there's nothing, but obviously you one maybe in this one you are nothing. Let's see in this one. One query is found. This is going on, trying to pick it. So still going, that one is still going on. And let's see this one, mutation. Okay, so this one has no mutation, and you have to also remember that these are only 15 nucleotide, 15 to 20 nucleotide base long. So if we are not finding a mutation in this region, there can it should not be a problem. But there are several ways of identifying in non-coding genomics mutation. And if C-Bio portal is not primarily focusing, then we have to change the database. We have to do some other strategy making. So yeah, but if you know the strategies, we can do it. So depending on which strategy you are working on, why you are looking for, one can do it. So one way is that base slide base or slide base. So there's a way slide base. Slide-based database is providing microRNA from enhancer promoted. So this is where you can have a look. So if you're looking for human microRNA activity in human microRNA activity in different cells. So now I am here. So there are different so the results for all. Now they have different microRNAs, and then you can download different microRNA, which cell they are involving, which okay, so which cell they are involved in, and then which expression pattern it has. And here you will also get you can go and discover it in UCSC Genome browser. So it will allow you to go to a UCSC Genome browser, and also some other you can get the sequence. Just get sequence. So it will fetch you data from the different sources, and so on. So you get a lot of information from here, and then suppose you think that your microRNA is binding, or you can have a browsing experience. Also, microRNA track is coming. Okay, view the track so that. Okay, so a track will open actually now. Let it open. Yeah. So here is the microRNA, and here is the protein. And these are the expression changes flanking the protein. So you see that you can map directly. And now if your mutation was in between here, this will definitely have implication. So that's when you and I and everybody has to look carefully what we are doing. And then all these 1857 microRNA are listed here, and which are many of these their finding are experimentally validated. So therefore you need to change the strategy. If the cancer database is not directly giving you go to other database, fetch it and match it with the cancer data. Okay, with this, if you have another question, I can answer, or else we can have a final.
Sir, we can have one more question. Can you please suggest open access genome data set for breast cancer?
Oh, yes. For breast cancer, this database is we are taking from breast cancer. We have taken the data set of. Okay, so we are working on breast cancer, and we, as a standard data set, we are taking cBioportal. Okay, so then I'm just taking the cBioportal. cBioportal. So the cBioportal has one of the largest breast cancer samples available. So yeah, so any cancer, the cBioportal is currently a good one. So there are 18 studies. So you can select all these studies and explore selected studies. So we just combining the information. So it will take some time and you'll get all the information. So and no bigger than this this. So recently we have provided this data to our collaborators. So this is 9,150 in samples, 9,557 samples, and then 9,072 samples have mutation in the genes, and you will get the number of genes here. Survival profiles, all no not bigger than this you can get for, and then your microRNA expression profile, and all this you have to click and start go inside it. So as you see, this is still working for the gene profile. So we were doing the cancer gene mutation in breast cancers locally in India, and then we compare this with the profile we received from we received uh, uh, from our local with the Icgc with this TCGA, or it's primarily TCGA data, but actually it is got now. If you are collecting from here, I call it cBioportal data we have taken. So this is working, but once it is working, then you will see the which gene is mutated and you and in how many samples. So we have used this very extensively. Result we have provided to our collaborator last week only. So and it's still study is going on. So you can use this for the breast cancer. So it may be coming. So another question. So when it comes with so is there any other question? So I'm going back. It's still working. So you can also you just saw how I did. So you can also go and do it, and you can then classify different type of cancer, breast cancer, and all type of different studies.
Okay, sir. So I feel we do not have any set of questions remaining.
Okay.
So thank you very much, sir, for such an informative and explanatory and resource.